bring you greetings on behalf of President Daniel Lehman and our college community. And I want to pause for a moment to ask the question, who would have thought? Who would have thought that a year ago, or even at the beginning of this semester, that we will be having this important event, the annual Student Research Advisory Board Scholarship Showcase virtually. But I want to say thank you so much for the work all of you have done to bring this event to us and to continue the important work of faculty at our institution and certainly our students. Here we are, guided through the wonders of human creativity. We are blessed with the technology platforms that allow the opportunity for us to continue to engage each other. And I want to congratulate all of our student presenters for their accomplishments. 30 of you, as I understand, were selected to present at this year's conference. And I had a moment last evening to look at the program. I saw an impressive cast of titles, the work you are doing representing an emerging body of promising scholarship. And I want to say thank you to all of you. One of those presentations titled, Why Worry About New Treatments for Cerebral Malaria by Jimmy Sanchez, and the other one, Relationship Between Acculturation and Attachment by Nabonu Diaby, were some of the presentations that took place this morning. I want to thank all of the faculty sponsors for guiding our students. What you are doing at Lehman with our students is what scholars do across the globe, to foster curiosity about the world, to teach our students not just what to learn, but how to learn, how to formulate new questions, how to make discoveries about the unknown, and how to find solutions to urgent human problems, urgent human challenges. That role is what is encapsulated in the work of our keynote speaker this afternoon, Sarah Pearl Eggenduff, who will be receiving, as I understand, her PhD in Earth and Environmental Sciences from the CUNY Graduate Center this semester. Congratulations to you, Pearl. In August 2020, she will begin a position as a postdoctoral researcher at Cornell University's Atkinson Center for Sustainability. As a master's student at Brooklyn College and a PhD student at the Graduate Center, Pearl has conducted research on urban soil containment and nutrient cycling and is interested in the ways that newly constructed soils can support climate change mitigation and environmental justice. She draws from a range of biogeochemical and participatory action research methodologies to co-create research with experts outside of academia at a variety of scales. She has conducted the first pilot study for the New York City Mayor's Office of Environmental Remediation, the unit that manages Clean Soil Bank. And she has also been involved with numerous follow-up studies, notably through the Carbon Sponge Project, the Just Soil Project, and the East New York Farms Healthy Soils Initiative. She has a bachelor's degree in education studies from Brown University, and she also holds a master's degree in teaching from the University of Washington. And Pearl's work has drawn from her diverse teaching experience, where she has spent some time teaching at both Brooklyn College and Queens College. As a postdoctoral researcher, her work has been based in our city, the city of New York, where she plans to continue the work in partnership with other scientists, city agencies, and nonprofit organizations, including artists, composers, composters, gardeners, and young people, to both study and apply best practices for systemic contaminant mitigation. She also has done some work on waste reduction, greenhouse gas sequestration, as well as food justice. And the list goes on and on and on. Please join me in welcoming to Lehman College our keynote speaker and soon to be Dr. Sarah Pearl Eggenduff. Let's give her a warm welcome. Sarah.
Thank you so much, Dr. Nwasu, and thank you, Dr. Sisselman and Dr. Hirokovich, and all of the students who've presented research for this symposium. It's really an honor and a privilege to be here, so thank you so much.
Welcome.
Let's see, can you all see my screen now? Great. Okay, so today I'm going to talk about building new soil, and we're building this soil for healthier urban communities as well as our healthier urban environment. So I'm going to talk about, first and foremost, some acknowledgements. Then I'm going to talk about lead in urban soil and what we can do about it. I'm going to talk about carbon sequestration in soil, what it is and how it works. And then I'm going to show you some examples of these projects where we're constructing soil in New York City. As Dr. Nwasu mentioned in my bio, I work with a number of collaborations. Some of these are the Carbon Sponge Project, Just Soil Project, and networks at broader scales. So talk about connecting all of this across time and space. First, I'd like to make an Indigenous land acknowledgement, and this is becoming more and more common, and this is certainly a common practice for Indigenous communities. So I was born and raised on the unceded land of the Wappinger and Lenape peoples, known as the Bronx, and I'm presenting to you today from unceded land of Canarsie and Lenape peoples, known as Brooklyn. I want to begin this presentation by acknowledging that we in New York City are in Lenapehoking, the Lenape homeland, and by acknowledging the Lenape community, Lenape elders of the past and present, and future generations to come. Many of the institutions we are part of were founded on exclusions and erasures of Indigenous peoples, and this need not be the case anymore. Given that CUNY strives for justice and accurate, accessible education, I invite all of us to commit to the process of working to dismantle the ongoing legacies of settler colonialism in all of our vital education and research endeavors. In addition to acknowledging the Indigenous communities that we are essentially connected to, I want to thank the essential urban growers across the world, urban and non-urban growers, but especially in the urban environment, community gardens and urban growers exist at this intersection between urban sustainability and environmental justice. So some of the work that they do to increase personal, community, and environmental health are providing access to fresh produce, food sovereignty, physical activity for participants, space for community building and cultural exchange, use of abandoned lands, reduced waste, improved air and water quality, and so many other environmental amenities. Urban growers also help us limit this legacy of lead that we have in soil. So what you see on the right is a map put together at the lab at Brooklyn College, and people sent in soil samples, but it's not a systematic map of the entire city. But what you see are zip code areas, and everywhere where the median value of samples sent in is above 400 parts per million, those are areas that are red. When the median value is between 150 and 399, it's yellow. Green is below 150, and no data you see is throughout the city. So we see such a large concentration of red areas in Brooklyn because the lab was in Brooklyn College. We see lead all over our city and many other cities. Fun fact I'd like to share, did you know that 36% of New York City's surface is covered with soil? We actually have a lot of soil in our fine city. And although no systematic map of New York City soil lead has been created, it's very common to see these high concentrations. Something also worth noting is that naturally occurring background levels of soils that should be unaffected by humans may have 20 to 60 parts per million. So this is common, and lead is an element and can't be broken down or effectively removed from soil, which I'll talk more about later. So this puts not just gardeners, but really all urban residents at risk for exposure, especially kids. And soil lead has also been linked to environmental justice issues. So here you see maps of Oakland, and with many other environmental issues, we see low-income communities of color being more exposed and more at risk to soil lead. So here you see high soil lead, which is in the bottom left. Again, red areas are greater than 400 parts per million. You see 20 up in the lower lying hills, or 20 to 80. And this is correlated with communities of color. You see more communities of color in the lowlands, high rates of poverty, and high blood lead levels. So there is this argument that soil lead can be a contributing factor to blood lead burdens that are found. And these unjust patterns occur in other places, and they must not be allowed to continue. We can recognize this and do better. I wanted to also mention that lead is something I talk about a lot. It's very common. We've used it in all kinds of manufacturing and industries and paint and gasoline, but it's not the only contaminant of concern. It's also relatively easier to test for lead with a rapid screening device that shoots an X-ray at your sample called an XRF than it is to test for things like PAHs, polycyclic aromatic hydrocarbons. You need more in-depth analyses through gas chromatography and processes like that to test for these other contaminants. So there's many, traffic, lumber, burning waste, manure, sludge, petroleum spills, all kinds of activities produce contaminants that can be found in soil. So lead is common and easy to test for, but it's not the only issue. And we refer to lead for simplicity, but other contaminants should be kept in mind. And the other contaminants are not necessarily correlated with the lead levels because lead was used so commonly. So what do we do about it? I can't really see everyone's hands, but this is a very common question. Can we remove lead from soil? And can plants help us do this? It's known as phytoextraction. I don't know if people have heard of this. Often, I don't even see a lot of faces here, but I often get some nods. People think that you can plant sunflowers and they'll just magically suck up the lead. It doesn't really work, break it to you. Not safely, not effectively, not on human timescales. And there was some research done, especially in the 1990s, that showed that if you add some chelating materials to soil, you can make the lead more mobile. It's organic molecules that bind with it, and it can be more mobile, and crops and plants can take up slightly more than they would otherwise, but then the lead is more mobile in soil and it can leach down into groundwater, creating a larger issue. So this is not an effective solution for mitigating exposure to lead in soil, but this is also good news because most plants take up very limited amounts of lead through their roots. So most are usually safe. The important thing to do is make sure you wash off any dusts that land on the surface that could. Okay. Could you add amendments to soil to make the lead less toxic or less bioavailable? And there's a lot of amendments that you can add. Here's big pictures of compost. A lot of amendments are great for soil, but they don't necessarily address your lead issue. It's tricky. So testing disputed for this. Adding amendments with more phosphorus, which compost tends to have, can make elements like arsenic more available, even if they make lead less available. So amendments are great in general, just they won't necessarily address your issue. Okay, so can you dig it up and haul it out? Certainly, if money's not an issue. And I don't know if you're familiar with what's going on in Red Hook, but there were these ball fields in Red Hook, Brooklyn, that were built on top of a former lead smelting factory. Generations, even NYCHA communities are right there being exposed for generations, and they've been closed since 2015. It's now been 5 years. They started remediation, and it's costing $100 million to remove the soil. So because it's so time consuming and if you want to remove oil, and also you're limiting people to that green space, which is really essential for health, the best thing you do is cover it. And so here you see an image of a garden plot being covered with a permeable landscape fabric, and then you can place new soil on top. But when you do this, so I listen to a lot of researchers who advocate new soil, covering what's below, but where do you get new soil? So that brings us to a project I started as a master's student at Brooklyn College, which is known as the Clean Soil Bank. And this was created by the Mayor's Office of Environmental Remediation. And they oversee sites throughout the city. And often what they find is that if you dig deep down below the surface, which our surface may be contaminated with activity, when you dig below, especially in Brooklyn and Queens, there's a lot of these glacially deposited sediments, which is mostly like sand. Brooklyn and, the Bronx and Manhattan are more crystalline bedrock, as you see the outcrops up near Lehman and, the Botanical Gardens, Bronx Zoo, Central Park. When you dig down, you may not find sediments. Queens, you do. And a lot of these sediments deep below the surface have never been touched by human activity. They're perfectly clean. So when they've been thoroughly tested and found to be clean, this office was wondering, well, what other purposes can they be used for? They helped build Brooklyn Bridge Park. They've been distributed around the city, but they were never used as a growing medium. And what we found is that if you add compost, which in our first was provided by the Gowanus Canal Conservancy, that provides the organic matter you need for growing plants, nutrients. We brought those to various community gardens in Brooklyn. We studied these soils, and we found that they produce a viable growing medium. There are some images of the beds. And we found that they are indeed viable growing media. The soil parameters are suitable for edible crops. And we found that at least 33% compost was adequate for comparable yield to what gardeners were otherwise growing in. Crops dominated, and these soil metal concentrations remain very low, even when surrounded by contaminants, at least in particular in one garden. But here's some pictures of these soils 3 years after they were constructed. And I don't know how familiar you are with looking at soils, but it doesn't look like most soil you see. It still looks like a sand and a compost mixture. These are those 33% mixtures by volume. So we have all of these other research questions that we want to ask. We know that plants will grow in it, but they're not yet looking like naturally occurring soils. So we want to know what biological, chemical, and physical processes are going on in these newly constructed soils. What's going on over time and what's going on in different spaces. And how much carbon is being sequestered in the process. How can we even optimize those conditions? So as I've mentioned, the sands that we start with lack all organic matter, all organic carbon. You add a little bit of compost, but over time we want to see, can more carbon build up? So now I'm going to switch to answering some of these questions. So carbon sequestration, especially in soils, is this idea of removing atmospheric carbon dioxide by plants. This happens through photosynthesis. Plants are doing this all the time. And they're storing it as carbon in soil, either as soil organic matter, SOM, or soil organic carbon. And I'm not going to go into detail of all of this flux diagram, but there's all these new ideas that have been emerging, whereas a traditional view really saw this soil carbon specific compound, that it was really a biochemical transformation that took the carbon from plants and into a more stable form. And actually what we're seeing now is that it's soil carbon is more a result of the ecosystem processes around and how protected that carbon is from disturbance and decomposition from microbes. So basically, this again is a sort of an up-close version of what's going on with soil carbon sequestration. The old view is that carbon, plants photosynthesize, they take the carbon dioxide out of the atmosphere, their leaves drop, and this surface litter was really important for, making soil carbon. And then again, some would be decomposed, released to the atmosphere. But what we actually find is that the surface litter is a relatively small contribution to soil carbon, and it's really what's coming out of plant roots. And it's really a function of how disconnected those molecules are from decomposition and microbes, how those molecules sorb and desorb off of soil particles, how they freeze and thaw, and that deep soil carbon, which really can be stored over long periods of time, is a result of these other processes. I want to mention on this past image here, again, the soil reservoir is really large. These are in petagrams or gigatons of carbon. So the atmosphere is 829, where soil has almost 5,000. So it's a huge reservoir, and we're releasing a lot of soil carbon every year, and we can store a whole lot more of it. So how do we understand this process, even in urban environments? Again, there's these inputs, depends how the soil carbon molecules aggregate, and we create there are stable microaggregates that form with these organo-mineral complexes, encapsulate and protect carbon against microbial activities and decomposition, and then they can translocate or move down into the subsoil. So soil organic carbon moves away from these zones of disturbance from anthropogenic activity tilling the soil, for example, compacting it, and other erosion. So there's all these ways that we can get more carbon into our soils, and there's ways that it can be released. So I've already sort of mentioned that it's microbial respiration or decomposition, where these soil organisms eat these carbon-containing molecules and break them down and respire them, much the way we do. We exhale carbon dioxide or methane. So there's these outputs of decomposition, respiration, mineralization, turning it into an inorganic form, and oxidation, releasing to the atmosphere, leaching and erosion. Carbon can be lost from soils. And we have much more carbon being lost from soils over human timescales, 40 to 90 gigatons, which many researchers argue could be replaced. And this can greatly offset the impact of anthropogenic climate change. It is not the only solution that should be approached, but it's a simple one if we already need to be building soils to support food justice and food access and all of the benefits of urban greening activities. So soil organic carbon depletion is a reversible process, and we can restore more to soils. So again, these big ideas are that carbon sequestration is dependent on these environmental factors and soil structures. And our questions are, how do we manage and support soil in aggregating these conditions, particularly in the urban environment? And we notice that increases in carbon storage occur quickly with carbon depleted sediments, like the ones we have. So we have this very large supply of these subsoil glacially deposited sediments. And so we want to know what we can do to optimize these conditions and increases over time. The big idea, again, is to cover contaminants, limit the exposure, build new soils on top, plant perennial crops ideally, add compost, these soils, and in the process, we're creating jobs and education and creating a healthier environment. So here's some pictures and examples of the projects in which we're doing this and asking these questions. This is images from the website for the Carbon Sponge Project, and this started at the New York Hall of Science in Queens, which if you haven't been there, growing up in the Bronx, I never went to the Queens Hall of Science. It's a fantastic science museum. Highly recommend it. And these plots are still on site there. So we got to go interborough sometime, well, maybe when the pandemic is over, but and so we started with the same amount of compost and had the different planting combinations. And here's images from the Just Soil Project, which is done with the NYCHA community, also in Queens, in Astoria. And so we're studying these soils with young people and asking a lot of these questions. So here's sort of an image from that where we're constructing soil for environmental and social justice, using these glacial sediments, using compost, studying them with researchers and youth and community members from a NYCHA community. And here's what it looks like when these sediments are being dug up. So here was what they call a generating site in Queens. And again, these sands were left here by glaciers some 20,000 years ago. So they're loaded up in big dump trucks, brought to a site, and then we mix them by hand. We carry the buckets and we mix them with compost here at the 33% ratio, place them into beds, and then plant different crops, and we have these productive soils. Here we are with the Just Soil Project with youth, starting seeds, building beds. We got our pile of sediments too, and we had to bring them in wheelbarrows, mix them. And what we have here is a layer of sediments at the bottom so that as we core these soils over time, we'll see what is leached down into this layer. And then we put the compost mixtures, and compost is very light and easy to be picked up by young people, mixed in wheelbarrows, placed into beds. And then we did different ratios of compost here, planted all the beds with the same number and variety of crops, and had a very productive first growing season as well as some subsequent ones. We weighed our yield, had a cookout, and kids were really enthusiastic about greens in ways that I hadn't really experienced before. So it's a really fun and meaningful way to engage in science and personal and community health. And here we are at the lab with some people associated with the project. This is at the Advanced Science Research Center, analyzing these soils for all of these various microbial, physical, and chemical properties. So I just want to mention on a larger scale, we really need to get everyone on the same page about how we deal with this. I often present this information. I wasn't aware of what's going on in our urban soils before I really got into this work. And so it shouldn't just be limited to us few scientists or few project partners who understand this. So since 2016, we've been gathering about 20 plus organizations, asking how we can build a coalition to support this critical soil education, understanding lead and best practices, and connecting with K through 12 garden education networks. And here's a sign of revised best practices of what you can do to limit exposure to lead in soil. We also have more info sheets, and also doing this research. So these follow-up projects like Carbon Sponge and Just Soil came out of this network. And this also was the beginning of building a clean soil distribution network. And I don't share photos here, but the first pilot distribution is happening with East New York farms, where local gardeners are getting more access to this soil. And in some, even beyond, hundreds of regions have been mapped for soil lead across the US and abroad. And here's different areas in the country that where estimation of lead exposures were produced. I've only found 23 reported articles on soil lead interventions. So we map the contaminants everywhere, but we're not documenting what people are doing to limit exposure, which gardeners and people who are building new soil are doing all the time, whether they recognize it or not. So I think we need to keep building our local partnerships to understand these interventions and the good work that essential urban growers are doing. And we need to rely on our academic resources to do this, to study our local particularities, support these efforts, and ideally build our connections more distally across our country and across the world. And I've had the privilege of working closely with researchers in 9 cities, some abroad, and I think we can keep expanding on this as we move over time and space to create new soil. So here's sort of an image of over time and space. I see this as working on them. And asking these questions is how can affected communities, and not just the researchers, construct soils to sequester lead and carbon? And how can community and scientific partnerships further these efforts to reduce waste, reduce contaminant exposure, and promote environmental justice, food justice, and all the benefits of urban green space? And so far, I've been looking at this on the small scales, building new soils with examples like Carbon Sponge and Just Soil. On the meso scale, sharing knowledge through this legacy-led coalition. And on the macro scale, creating interagency collaborations, more policies, and seeing New York City as a model city. We can do it here, we can do it anywhere. And so my question for all of you is, how might we work together? I'm gonna stay involved in this work based in New York City, not through CUNY, but through Cornell, but I'm gonna keep expanding on this for the long term. And if you have any interest in joining in, thoughts, ideas, questions, please feel free to stay in touch. Here's my email. And thank you so much.
Thank you, Pearl. That was wonderful and so interesting. We have one question from we actually have a couple of questions in the chat box if you'd like to read them, and feel free to answer if you have time.
Do you look at climate change and how that might affect carbon in the soil? I believe with climate change, there's an increase of carbon in our carbon cycle. Absolutely, there's a lot of different predictions for what's going to happen with it. We know we're going to have more carbon dioxide in the atmosphere, more greenhouse gases. That is known. Exactly how much and exactly when or what people debate. But with more carbon dioxide in the atmosphere, there's different hypotheses about what's going to happen with soil. Some people believe that more carbon dioxide in the atmosphere will promote more plant growth, which is good. So there's potential for more. If there's more plant growth, there's potential for more exudates coming down from the roots and more soil carbon in soils. But there's other estimates that say with increased temperature and warming that increases microbial activity, you can also have more decomposition of that soil carbon, even more release to the atmosphere. So increased atmospheric carbon could increase soil carbon, which could be a negative feedback, or it could increase soil carbon decomposition and be a positive feedback, increasing warming. So there's a lot of unknowns, and what we're trying to study is what does increase the soil carbon? Are there methods that we can do that effectively, especially in the urban environment? So still a lot of unknowns and what's going to happen, but I think we can try to make some interventions before it's absolutely too late. Is the soil dangerous to animals that live in it? Are factory farms looking into creating a new soil? Two great questions. So if soil is contaminated, as I've mentioned, much of our urban soils are, that can certainly be harmful to other animals. There are stories of cats in the urban environment who've been contaminated from lead. And so it affects humans, it affects other organisms as well. There's actually a lot of microbes and plants that they have ways to protect themselves. They've been around a lot longer than us mammals. I think they know what to do, how to, not take up these toxic elements, but it can certainly be harmful to other organisms. Are factory farms looking into creating a new soil? So we've got urban soil contaminants. There can also be rural soil contaminants. Often rural soils and factory farms are contaminated by all of the waste that's produced there in one local area. So that's tricky. I would like to think that they're working on this, but I can't guarantee it. I think that often in those situations, there's a lot of unsustainable conditions, and the goal is profit over the health and well-being of the organisms being cultivated there. So I think it's, I go to soil conferences with big agricultural producers, and they're still using a lot of pesticides, which may also have other contaminants. And they're starting to get more onto the climate change, right? Trying to store more carbon in soils, but building soil health in terms of contaminants and exposure to toxins is lagging behind. Nitrogen. Okay, wait, did I go that? What about nitrogen in soil? Plant growth is limited by available nitrogen. Absolutely. So this was a very short, condensed, limited presentation here. There's no way to look at soil carbon without looking at nitrogen. They cycle together. Nitrogen is the next highest abundant element in soils and in microbes. So we always look at the carbon to nitrogen ratio, and certainly plant growth is limited and by available nitrogen. And and also nitrogen can not only in the form of nitrous oxide be a powerful greenhouse gas, but can also be a water contaminant in the form of nitrate. So we're looking at all of these dynamics together. So as that data is becoming available, I'll show you what we're beginning to find in terms of how these elements cycle together. You can't really ignore that. So I talked about lead and carbon, but they're just sort of signifiers. They're not the whole picture. Any recommendations to sites to learn more about urban soil? I have no background in soil science, but it is interesting. Well, I agree. So good sites, if you start searching for Cornell Healthy Soils, Healthy Communities, that's a group I've been working with, and they've got a ton of resources available online, especially around soil contaminants. You can check out the Brooklyn College Urban Soil Lab, one of the labs where I've been working, and there should be some links on there. Also, under normal times, that's a place where you can mail in your soil samples and get them tested. They also always accept interns and volunteers, so it's a great place to actually physically go when we're able to, and get some real hands-on learning about soils. So Cornell, Brooklyn College are great places to start. What kind of economic model / approach is being looked at to fund the cost of making what are essentially large-scale raised bed construction projects? Between transporting the sediment, lumber costs, and labor costs, the price seems potentially limiting for local urban gardens and small-scale groups. Absolutely. So this is something I hope to look into more, the feasibility and the economics of this as a postdoc researcher. First, I wanted to see, as a PhD and master's student, does it even work? Can this even happen? We see that it can, but I think it's true. Eventually, we're going to need ways to fund this, and it could be city funded. So the city agency that I've worked with, the Mayor's Office of Environmental Remediation, they have this abundance, they oversee the transfer, this abundant supply of sediments, and the city's been somewhat reluctant to use them on a larger scale. But now, even over the pandemic, the Parks Department just received 7,000 cubic tons of sediment. So this has been years in the making, since 2013. It took 7 years, and the Parks Department said, hey, this material is clean. Oh, look, it works as a growing medium. They just accepted this. So that's going to hugely offset some of their costs. maybe through that process, there could be some funding and other economic models. We need more green jobs. But something I meant to mention too is that the mayor's new budget just canceled all city composting efforts. So this is really a devastating occurrence, and I don't have the links immediately available, but I'm looking to call our local council people to try to reinstate composting. This is both the city's brown bins, which were problematically only placed in a few neighborhoods, but also all of the compost drop-off programs. I don't know if you ever go to farmers markets or see people that collect compost, and they do this through NYBG and other places in the Bronx, all over the city, completely cut now. So yeah, I see some wows. Thank you. It's devastating. So we need to call our council people and tell them we need composting. This is essential for the current and future, health and environment of our cities. So I do think there's a lot of potential for the city. Oh, so there's bills that are looking to be passed to make composting mandatory. And so if the city funded mandatory composting, and there's been some pushback from mandatory soil testing, but there could be, funding to support access to new clean soil. And we have the space and the ability to do this. So we should look. I don't have answers on what the economic model actually looks like, but I hope to create that soon. And if you have ideas and want to reach out, please, I'm open to that. How do you know if your soil is safe to plant a vegetable garden in? First and foremost, so the labs are closed right now. You can't send a soil sample in. But if that soil has been there a very long time and it's in the city, there's a good chance there's some contaminants in it. My backyard in Crown Heights has 2,000 parts per million of lead. And I saw on the back of my building, there's peeling paint. So often it's the highway traffic and the street traffic that from, when we had leaded gasoline that contributes contaminants. But if there's peeling paint, if there's infrastructure, there's a busy street, good chance you've got it. So the best thing to do if you can't test is to bring in new soil. If you don't have access to new soil, or there may still be some distributions or pickups from East New York Farms. So if you want to get some, you can also reach out to me about that. So the best thing to do is get new soil. But if you can't, then there's some important things you can do, like don't plant rooting crops, limit your vegetable crops, plant mostly fruiting crops, tomatoes and peppers. They have all these barriers. They won't take it up. Always use mulch, prevent dust and splash, and wash your veggies. And I found, another research, which I didn't present here, but even in highly contaminated soil, if you wash them very, very carefully, you're largely safe. But to be extra safe, grow in new soil and limit splash and wash them. And under normal times, send your samples into Brooklyn College. That will help too. Okay, let's see. The Bronx has several concrete and soil recycling centers in the Yankee Stadium, too, that add a lot of dust and other pollutants to the air in the neighborhood. Is there a way to scale up your work with these sorts of companies in a way that might mitigate their local impact? I love that question. Want to work together on it? Let's do that. Okay, I mean, dust and exposure is huge. That's really what, breathing in the dust, and then that's what lands on our crops, and that's what even could potentially recontaminate these new soils. You can bring in a clean soil, but if you've got a big construction project, distributing contaminated dusts, that's an issue. And so there's literature that show that that in the drier summer months, you have an increase in dust in the atmosphere, you have an increase in contaminants, and that can recontaminate clean sites. So we've got access to these materials, and we're working across the various boroughs. So the more locally based and connected projects are in the community, the more effective will actually occur and reach their goals. Please reach out if you're interested in that. Can you tell through observation? It's very hard to look at a soil and see if it's contaminated. So some of the work I've done, as you see with those newly constructed soils, they don't look like a healthy soil, but they do support plant growth. But I've had comparisons with very contaminated soils that had been used for years and years, and they look beautiful and had this incredible plant growth, and they had like 800 parts per million of lead and 600 parts per million of arsenic. They're contaminated, looked great to the eye. So You can't really tell just by looking. You can tell by thinking about what's gone on in that area over time. Has new soil been brought in or not? And if not, it's a good chance you've got contaminants. Using legumes as a choice of plant along with nitrogen fixing bacteria, always recommend. So legumes, right, have this amazing symbiotic relationship with nitrogen fixing bacteria that are able to take that essential plant nutrient out of the atmosphere and bring it into soil. So we always plant legume cover crops over the winters to add more nitrogen to the soil and keep it healthy, limit exposure to dust, limit the soil from moving away. So as much as you can, planting those types of crops and cover crops, even not in the growing season, highly recommended. So you don't see pictures of our cover crops, but that's always, I didn't emphasize that, but that's always there. Did I miss any other questions? Anything else?
It looks like there's a couple. Can you use legume as a choice of plant along with nitrogen fixing bacteria? And can you tell through observation?
You know, I think I just answered those,
Yeah,
but I can.
I think it was pretty, I think we got those, right?
Yeah.
Okay, cool. Anybody else? All right, well, this was really quite interesting and amazing. Thank you so much. Really interesting to see what is going on in our soil and how that affects all of us in the city. I really appreciate you doing this for us. You're getting lots of Zoom claps and giving you a real clap. Thank you so much. It was
Really my pleasure. So again, I shared my email. I'm happy to stay in touch about all of this.
That's great. We will make sure to share email with everyone to make sure that they have questions they can follow up with you.
Wonderful. Thank
you
you
again. Okay, so we have another 4 or 5 minutes for a brief stretch before we get back to our student presentations. So why doesn't everybody get up and breathe and do a little movement just so we're not all sitting so much. We've been sitting all day. And then we'll come back in about 5 minutes and we'll get started with our next student presentations. Pearl, you're welcome to hang around and view more of our student presentations. They were absolutely amazing this morning and I'm looking forward to more this afternoon. Okay, everyone, I'm going to welcome us back one more time. For those of you that have joined us later, I will do my brief welcome message one more time for all of you, and then we'll get started. So good afternoon and welcome back to those of you who have been with us since this morning. Thank you all for joining us today. My name is Amanda Sisselman-Borgia, and I am chair of the Student Research Advisory Board. I'm getting tongue twisted now. I would like to congratulate all of our students who are participating in the Student Scholarship Showcase this week, both live and asynchronously. I'm impressed with the number of students who came together to showcase and present the amazing work that they are doing, despite the difficult times that we are in. I think this speaks to the resilience and the incredible tenacity of our Lehman students. Thank you also to the faculty sponsors who have been supporting this great work. I know you do a lot in the background to support our students. I have been participating in and viewing the asynchronous presentation since yesterday, and I invite you all to do the same over the course of the next few days. I'm impressed with the quality of the work, the diversity of topics, and the importance of all the work being presented. We had an amazing morning and heard from so many interesting students and topics, and I'm looking forward to hearing this afternoon's presentations live. So our first presenter is Eris Bisal, and Eris prefers not to be recorded. So I'm going to pause the recording and let Eris begin presenting. In the meantime, Eris, we had one other question for you. Why did you choose this study, from Nina Albassam?
Well, I hope Dr. Chang's not listening, but it was kind of given to me. But I do find it interesting, and I do want to work. Oh, hi, Dr. Chang. Well, that's why I studied it, because it was given to me. But I mean, if I were to pick, I would pick this one because, yes, I do just find it interesting.
Excellent. Thank you so much.
Okay, so now.
Okay, Mrs. Ready. You're ready.
Yes, I'm just gonna share the screen right now. So
we'll give you a reminder when you're getting close to time.
Thank you. Go to Zoom, share screen. Okay, everybody can see my my PowerPoint?
We
All
can.
right. All right, thank you. Hello, everyone. My name is Musa Payinke. I am a senior at Lehman College. I've been doing research with my PI, Julio Delgado. And so today what I'll be talking about are the effects of angiotensin II in the development of cerebral malaria. So to start off, this is going to give you some background information on malaria and cerebral malaria. So what malaria is malaria is a life-threatening disease that's transmitted by the bite of an infected mosquito known as the Anopheles mosquito that has pulled in 228 million new cases and over 400,000 deaths 2 years ago in 2018. Cerebral malaria is a complication of malaria caused by the parasite Plasmodium falciparum. Pretty much what happens is you have these parasite-filled blood cells attached to your blood vessels on the brain, and this results in disruption of the blood-brain barrier. This also causes swelling of the brain with a high mortality rate. Cerebral malaria is one of the deadliest complications of severe malaria, with a mortality rate of 20%, even when treated with anti-malaria drugs. What this research wants to focus on, or the purpose of this research, what it focused on, was the effects on different concentrations of angiotensin II in an experimental mouse model of cerebral malaria. This animal model uses two different concentrations of angiotensin II. And we essentially use these two different concentrations of angiotensin II and measured them against a control group to see what were the effects of angiotensin II in the development of cerebral malaria. Pretty much the experimental model between these three distinct groups pretty much included 15 mice in each group, and within two groups, each were infused with a dosage of 100 nanograms per kilogram per day at 0.25 microliters per hour of 100 angiotensin, and the same with 500 angiotensin. Infusion of angiotensin began the same day as infection, and all three groups were tracked from the first day of infusion of angiotensin to an infection up until the last day each of the subjects within each particular group survived. So the hypothesis, as mentioned earlier, are does higher levels of angiotensin II reduce the impact of cerebral malaria? Some of the materials that we used in this research was the histology station, because pretty much what we wanted to do was we wanted to collect tissue samples from all three groups and pretty much go and measure the number of hemorrhages that they had, as well as the length of the hemorrhages. This would allow us to see, does angiotensin II have an impact on the number of hemorrhages that we can see between the three groups. We also use the rotary microtome to actually cut our samples, and then we use a bright field microscope to actually visualize and count the number of hemorrhages between our three groups. If we look at methods, I have this diagram, and I want to see if I can actually zoom in. Nope, I cannot zoom in. Okay, well, that's fine. Pretty much what this diagram is basically trying to show is that we started from day 0, where we infected all three groups with Plasmodium bergii ANCA. And pretty much what Plasmodium bergii ANCA is this is the parasite that would pretty much infect rodents. So this is the parasite that would give cerebral malaria to rodents. And we infected all three different groups, and then within 2 groups, we infused the angiotensin at the different dosages. If we look at results, we can see a few graphs. The first graph that we see is a graph that measures parasitemia. Now, the reason why we wanted to measure parasitemia levels is because what parasitemia is basically a quantification of the number of parasites that you can find within the blood. And so pretty much what we wanted to do is we wanted to look at the levels of parasitemia from the total number of days that all subjects lived, and we wanted to see that over time, as the parasitemia progresses, what are the differences between the groups that have angiotensin and the group that does not. I'm gonna try to exit out of presentation mode for a second because I want to see if I could zoom in and actually show you the presentation. I mean, the graphs. Okay. Okay, yeah, you guys can see the graphs, right? Okay, so this is the graph that pretty much measured parasitemia. And if we looked, we have a control group, our Angio 100 group, and an Angio 500 group. We measured it, pretty much the parasitemia levels by days. We see that pretty much all the mice within the control group. Okay, sorry, just wanted to check chat just in case somebody's got a message to me. Okay, so back to the presentation. If we look at the control group, we see that all the mice from the control group died much earlier than the mice in either the angiotensin 100 and 500 groups. We also see that not only did those mice survive fewer days, they had higher levels of parasitemia at a faster rate. So pretty much what that told us was that when you have angiotensin treated within particular groups and you don't have angiotensin treated within particular groups, you can see a difference in the parasitemia, and that difference is that infected groups without any form of treatment will develop higher levels of parasites in the blood at a faster rate, and they'll also survive fewer days compared to other groups that have some form of treatment of angiotensin 100. The next graph that you can see on the right is called the Kaplan-Meier graph. So pretty much what the function of the Kaplan-Meier graph is, it's a graph that we use in statistics to pretty much measure the lifespan of some group that we're interested in. And typically what we do is we aim to measure the lifespan of a group with some form of treatment that it's been given. In this case, what we wanted to do is we wanted to see what was the probability of survivorship between all three groups. If we look, you can see that the control group, color-coded in green, died around day 8, whereas the angiotensin groups for 100 and 500 had a much delayed number, had a delay in the day of death. If you look specifically at Angio 500, at the group of Angio 500, you see that some of the mice actually were able to live for the full total duration of the time that we recorded. Not only do you see that the control group died at an earlier day, you see that they had a much higher probability of dying faster, or in other words, they had a much lesser probability of surviving. when compared with the other two groups. The next thing that we're going to look at are box whisker plots. So pretty much what these box whisker plots are meant to do is they're meant to show us if there's any significance between groups, and specifically between the MENA groups. Now, in order to verify if there's any significance between the MENA groups, what you have to do is you have to run a series of tests. You have to run these parametric and non-parametric tests because of the fact that you have to ensure that you have some, you have to ensure that the significance that you're getting is accurate. So some of the tests that we decided to run were tests such as the Bartlett test and the Shapiro normality Wilkins test. And those are two pre-tests that you do to decide what is the statistical approach that you'll do to verify if your means are actually significant or not. So when I ran the Bartlett test and the Shapiro normality Wilkins test, both of my tests came out significant. This basically meant that I could not run any parametric test and not have to run non-parametric tests. The non-parametric test that I ran was the Kriskell-Wallace test, and that also came out significant. When you have a significant Kriskell-Wallace test, you need to run a post hoc analysis test. And pretty much once you ran the post hoc analysis test, that would verify if your significance is accurate or not. So now, if you look at the box whisker plot, for number of hemorrhages by group treatment, what do we see? We can see that there is significance between the number of hemorrhages between all three groups. We see that in the control group that has no treatment of angiotensin 100, there is a higher number of hemorrhages versus either of the angiotensin 2 groups. The reason why we have an asterisk there is because if you look at the legend, and I know it's hard to see. Yeah, I'm not going to be able to show that. But if you look at the legend, pretty much what the asterisk signifies is the level of significance that there is between the groups. So if you have an asterisk of 1, then that signifies you have a significance that's below 0.5 but greater than 0.1. And if you have 2 asterisks, that will show that you have a significance that's below 0.01 but greater than 0.0001. In this case, each group has 1 asterisk because their significance is below 0.005. If we look at the length of hemorrhages by group treatment, we actually saw that there was no significance between the average length of the hemorrhages, which tells us that infusion of angiotensin 100 and 500 may not have an effect on the length of the hemorrhages. And then last but not least, these are some of the stains that we got using the hemotoxin and eosin staining. So pretty much what you can see here is you see 3 photos. This first photo is a photo of the control group. And you can see that when you compare it to both of the other photos, the second one being a photo of the angiotensin 100 group and the third photo being a photo of the angiotensin 500 group, you see that there are far more number of hemorrhages prevalent in the control group, which is consistent with the graphs that we have here. Because pretty much what that tells us is that we do have higher numbers of hemorrhages by group treatment for the control group as compared to the other 2 groups. And we also have higher levels of parasitemia, basically higher levels of parasites in the blood for the control group compared to the other 2 groups. We also have a lower level of survival probability for the control group, while we have higher levels of probability, survival probability for the angiotensin 100 and 500 group.
Hi, just giving you a quick reminder that you are getting close to time.
Thank you. I'm gonna present this on the bigger screen. So to conclude, earlier studies on the effect of angiotensin 2 on cerebral malaria have shown that angiotensin 2 can protect against the development of cerebral malaria, because when we looked at the data, as I mentioned before, we saw that you had lower levels of parasitemia in the blood, you had a higher probability of living longer, and you had fewer numbers of hemorrhages for both groups that were treated with angiotensin 100 and 500 when compared to the control group. In the study, we basically wanted to explore these effects, and again, the data pretty much answered or showed that our hypothesis that if you use angiotensin to treat cerebral malaria, you can impact it by pretty much inhibiting or slowing its effects. And just wanted to acknowledge Joseph Racklin and Julio Gallego Delgado. Joseph Racklin, pretty much said what his occupation is. He's the director for the Laboratory for Marine and Estuarine Research. And Julio Delgado, sir, this is my PI. And he also teaches at Lehman University. And that's the end of my presentation.
Thank you so much, Ezekiel. I mean, thank you so much, Mausa. That's fantastic. And really wonderfully presented and really wonderful posters. Who has questions?
Yeah, I'm trying to exit out of the presentation so I could check the chat to see if there's any questions.
Okay, yeah. You have some like that was great chat, but if anybody has a question, just unmute your microphone or put it into chat. Mouse is getting a lot of love in the chat, which is lovely.
Oh, thank you, everyone. I appreciate it.
It was really interesting. I have to tell you, I'm learning so much today from all of you. You're amazing.
Thank you. Thank you.
If we don't have any questions, then we're going to get ready for Ezekiel, who's been waiting in the wings. And I want to give 2 points. If maybe what would make sense to sort of facilitate having questions when this person is done presenting is if you're listening, like as you're listening, maybe you can write down little questions in a notes app or on a piece of paper as you're listening, or you can just put the questions in the chat, right? The presenter will do their presentation, and then when they're done, they'll look over at the chat and answer your questions. It's a ton of information coming at everybody, and sometimes our questions kind of emerge at particular points. And if you want, one way to make sure that the person's getting some nice feedback is if you write your questions out in the chat as they come to you as you're listening. And then the other important point is that we want to keep the presentations to 10 minutes, which is tough because you're clearly doing some pretty advanced and deep research. But check the clock. So with that, I'm gonna welcome Ezekiel, and it's now 1:35. So at 1:45, we want to be asking him questions. And welcome, and we look forward to seeing you, Ezekiel.
Thank you. Can you guys hear me?
Yes, we can hear you, and we're gonna just continue recording since you gave permission to have your presentation recorded. So thank you. We're gonna keep that up.
Yes. Thank you, guys. So today, my name is Ezekiel Olumoide. I'm a junior, and I'm a biochemistry major. I'm working under Mora Swani, and today I'll be presenting mechanistic aspect of interleukin 24. So I'm just going to zoom in. Yes. So cancer cells, what makes cancer cells different from normal body cells is that they overexpress some genes to produce, to upregulate some survival and proliferation proteins that makes them grow continuously, and they lose restraint over the translational machinery, which is the mRNA to protein translation. So what we are investigating is a protein called interleukin 24, which is also called melanoma differentiation associated protein, and this protein is known to cause different apoptotic effects on cancer cells, and overexpression of this protein could not only inhibit the metastasis of apoptosis of cancer cells, but also inhibit other functions of cancer cells. And what is great about this protein is that it does not affect normal body cells. So that was one of the great things about this protein. So what we do know about cancer cells is that cancer cells are different in every human body and in every individual, and even in the same individual, it could be different in the sites, in the microenvironment sites. So, but what we wanted to know is that what makes them common, and we do know that what makes them common is that translational initiation, that all of them have to go through translation to create their protein. So what I wanted to know, our purpose of the study is to know if interleukin 24 affects the translation of some of these proteins that or oncoproteins that cancer cells use. So meaning like if interleukin 24 inhibit like a initiation factor, EI4A, then to produce less oncoproteins, and then that would lead to apoptosis and cell decrease, proliferation, and cell death. So what we did, our experimental methods were viral infection. That we took a non-replicating virus, adenovirus, and which contains interleukin-24 gene, and we infect it into the cancer cells. And this virus then releases the interleukin-24 gene, which gets transcribed and then get translated and also secreted. And this secreted gene can bind to the cell, either even in the ER of the cell or on a receptor on top of the cell, and also binds to other cancer cells and causes apoptosis. And to do this, we use cell culture. This is ELA cells, and this is MCF7. This is ELA cells are cervical cancer, and MCF7 are breast cancer cells. So we also use KRT PCR, we use Western blot, we use the luciferase assay, and annexing VIP binding assay. So our results, what we got was interleukin-24 inhibit the gene expression of 5' untranslated region. So this is the so what we do know is that all proteins in the mRNA, they have some secondary structure on that 5', the upstream of the the mRNA. So then we did luciferase assay to look at different mRNA structures and look at the effect of interleukin-24 on that translation. So looking at this, you could see that there's no difference between all these structures that don't have this loop in the mRNA. So you could see that there's no difference, but in this, there's a difference, which shows that interleukin-24 actually affects the cleavage of this loop. So we know that EI4A is a helicase, is like a scissors that cuts this loop into two to straighten it out for the other translational initiation factors bind to. So we treated interleukin-24 to different cancer cells and saw that it causes downregulation in all cancer cells of EI4A complex, which correlates with this data. So we also checked for, sorry. Okay. So we also checked for the if interleukin-24 induced apoptosis through this, and we saw that the black means interleukin-24, the white means the control group. So we compared and we saw that interleukin-24 increased cell death in all these different cancer cells from different sites, from melanoma, from prostate cancer, from breast cancer, and from cervical cancer. And it also increased the apoptosis of all these different cancer cell types. And we also showed that interleukin-24 causes the upregulation of caspase-3. Caspase-3 is a pro-apoptotic protein that cleaves different proteins, that degrades different structural proteins in cells, not just cancer cells, but also normal cells. So since we know that interleukin-24 decrease EI4A protein, then we decide to treat the cells with EI4A and see if there will still be, if there's going to be apoptosis or cell death. Then we did that and saw that EI4A rescues the apoptosis and cell death of interleukin-24. So that shows that for interleukin-24 to cause its killing effects, we need EI4A downregulation. This, yeah. Sorry. Close this, yes. So now we decide to look at different oncoproteins, CDC25C, CMYC, ODC, and S-linked apoptotic protein, X-linked inhibitor apoptotic protein. We started to look at the protein level, and we saw that treating them with interleukin-24 downregulates all these proteins. And all these proteins have, in the mRNA, they have the secondary structure, this secondary structure. So it does correlate with our data that interleukin-24 downregulate interleukin-24 mechanism of killing is mediated through EI4A downregulation. Then we started to, since we saw that it's decreased the protein level, we started to look at the mRNA level of all these oncoproteins, and we saw that there's no difference in the mRNA level of all these oncoproteins. Then we concluded that interleukin-24 it inhibits all oncoproteins through the translational level and not the transcriptional level. Then previously, we showed that interleukin-24 physically binds to sigma-1 receptor. Sigma-1 receptor is a chaperone protein that is useful for folding of cells and also secretion of most oncoproteins, and also checkpoint inhibitors like PDL1. So we showed that, partially we showed that sigma-1 receptor binds to interleukin-24, and interleukin-24 have an antagonistic effect on sigma-1 receptor function. Then we decided to see if sigma-1 receptor rescues interleukin-24 inhibition of EI4E if it is upregulated. Then we decided to compare. Then we use sigma-1 receptor agonist, which is SKF10047, which causes the increase of the function of sigma-1. And we saw that when we treat this cell with the agonist, the sigma-1 receptor agonist, there's no difference. There's no difference between it and sigma-1. And we also show that sigma-1 is actually very important for EI4A, for interleukin-24 inhibition of EI4A.
Ezekiel, I'm going to have to ask you. Oh, here you are. The conclusion slide. Perfect.
Yes, I'm on the conclusion. So we concluded that EI4A inhibits translation of EI4A, and also it does this through sigma-1 receptor. Then our future research is to look at how does it downregulate EI4A. Then we postulated these 4 apoptosis that is through prostaglandin, through a tumor suppressor, through miRNA, and through a champion-mediated autophagy. Then I would like to acknowledge my lab mates, Christian, Justina, Ilal, Gabriel, for the feedback. And also I would like to thank the Lehman College Department of Biological Science for the fellowship award, for the 2020 fellowship award. I also like to thank the National Cancer Institute for the grants that helped with this project.
Thank you, Ezekiel. That is such important work, and you are doing, potentially life-saving work to help people who are suffering with cancer. So we appreciate all the work that you're doing, and you should be very proud of yourself.
Thank you. Thank you.
Yeah. And if you look to the chats, you'll see some questions there.
Yes, I saw a question right now. Is it advantageous to inhibit oncoproteins in during translation? Yes, it is, because oncoproteins are generated from oncogenes. Oncogenes are mutated genes that causes cancer cell growth. So by inhibiting these oncoproteins, you are reducing the survival of all these cancer cells, that proliferation, and you are causing apoptosis. So it's a really good therapeutic method by just inhibition of oncoproteins.
And do we have any other questions? Really excellent job, Ezekiel. Thank you.
Thank you, Ezekiel.
Nice to see you.
Same.
Excellent. And so next up we have Kelsey Goon, whose paper is Not Just the Tibia Sidekick, Measuring the Relative Cortical Area, Osteon Area and Shape, and the Osteon Density of the Fibula. So Kelsey, you've said that you are, you consent to being recorded. So we're just going to keep the recording going if that's all right.
Yes,
And
that's fine. Thank you.
very nice. All right. Go ahead.
Just gonna share screen here.
Mhm.
Okay. Let me go to my, oh, of course. All right. Might take a second. Okay. Hello, everyone. My name is Kelsey Gounet. I am a biology student here at Lehman, undergraduate, previously an anthropology, biology, chemistry student as well. So now I'm minoring in anthropology. So the purpose of my research today is going to be using histological measurements in order to understand the relations that the fibula can be used for in the field of forensic anthropology. So I'd like to start by explaining some of the methods and why forensic anthropologists use these methods. So forensic anthropology uses histological methods, which is basically methods looking at the microstructure of tissue to determine things like age, sex, and stature. And oftentimes skeletal remains are used for this purpose. One of the ways to determine age using skeletal remains is by identifying signs of bone remodeling. Now, bone remodeling is the way in which our bones maintain and repair. It's a system of maintenance and repair. And in order to see a little bit of that, I'm going to direct you to figure 2B. You can kind of see it here. Here we have a drawing of a zoom-in image of our osteons in our fibula sample. So here you can see a Haversian canal, which is the empty space here. It's an empty vascular space. And here we can also see like a tree ring pattern. So I'm going to explain a little bit of that as well. So as we age, osteons accumulate. So because of this accumulation, we are able to use it in age determination using skeletal remains. So similar to a tree building rings, our bodies also build rings in the form of osteons from bone remodeling. So oftentimes in this field, the femur or the rib is used, and this is because it's very well researched and because it's also very accessible anatomically. If you think about the femur, for example, that is your kind of like, I guess I should just call it the thigh bone for those of us that aren't, bone histologists. And then the rib, of course, is in your rib cage area. So why do we want to use the fibula is a good question. We want to use the fibula because it's something that we can use in addition to the rib and the femur. The fibula, unlike the rib and femur, doesn't have as much gross morphological use. And by that, I mean, as a whole, it's not as useful for things to identify different characteristics of the body. So because of this, it's a little more disposable. So it might be a nice addition. And we have seen in previous research in 1965 by Dr. Curley that has asserted that the fibula may be the most accurate bone to determine age. So essentially, my objective in this experiment is to figure out whether the fibula is a viable addition to the rib and femur in terms of measuring things like relative cortical area, osteon area and shape, and the osteon density of the fibula. All right, and to do that, we would like to identify trends relating to age and age determination. So in terms of how we conducted this experiment, I used an image software called ImageJ along with a cortical area macro set in order to find the cortical area of each fibula sample. Now, our sample is 5 individuals ranging from age 44 to 96, and they are all females. So this means that all of our samples are in the postmenopausal phase, and that's going to come in a little bit later. So in terms of measuring our intact osteons, so one of the things we measured was the density of intact osteons, also known as OPDI. And I'm just going to zoom in here again. So you can see here our osteons are circled in these red lines. So the reversal line here on our osteon is going to be that edge that's circled here. So we're essentially manually tracing the reversal lines of each osteon to get the measurement of its area and shape. In terms of the cortical area, we simply, you can zoom in on this image, we simply traced along the inside of the bone, or I guess the bone marrow cavity, most people know it as, and the outside of the bone, periosteal bone, and did a simple subtraction to find the area of the actual cortical bone. In order to set up a criteria to trace the osteon, so you can see there's not much, there's a lot of different shapes present in these pictures. I had to set up a criteria so that it was very standard. So one of the standards was that it was 50% or more of the osteon had to be present in each of these grid squares. The reversal line must be present and visible, and that the Haversian canal had to also be present, Haversian canal being this empty space. And in order to basically make our research both easily understandable and quick, considering the crisis that we're in right now, we decided to use 50% of the fibula cross-section. And to do that, we used a grid pattern, a grid with a checkerboard pattern to obtain 50% of the cross-sections. And these are all cross-sections from the midsection of the fibula. So here you can see the grid pattern that we created and a zoom in of one of our grids squares, which is from B6. You can see the numbers in it here. So as a result of all of our collected data, we found some significant trends that matched with the previous research done on these topics. The main one we saw that was a significant trend was between age and cortical area. Just to remind you guys, this one. So the cortical area did decrease with age, which was to be expected. However, the age and osteon area and the age and circularity, which is from a scale of 0 to 1, 1 being the most circular, 0 being the most abnormally shaped, there wasn't a strong correlation, which is also to be expected based on previous research. The age versus OPDI, or osteon population density of intact osteons, was an increase with age, which was also to be expected. And we can see that here in figures 4A and B, as well as figure 3 here. So I would like to get into my conclusions. So our experimental values did follow the trends from previous research. However, we do need to keep in mind that we had 5 samples, which is a rather small sample. Therefore, our study, there needs to be a little bit more study into this topic to confirm. Our cortical areas did decrease with age, which was consistent with research that had previously been done. And typically in these types of studies, they would use the OPD, or just osteon population density. But due to our lack of ability to use the microscope to check the images of the slides, it was a little more advantageous to use the OPDI. And regardless of that, we did see confirmation in trends, or a correlation, regardless of the fact that we didn't include things like osteon fragments or drifting osteons, which would be included in the osteon population density. So in addition, I would like to acknowledge the donors of our slides. And I would also like to thank Dr. Amanda Agnew and Dr. Sam Stout for the slides that we were able to use that they created. Thank you.
Thank you so much, Kelsey. That's fantastic. Does anyone have any questions? I'll leave that for a minute if people want to gather their thoughts and ask a question on Kelsey's research. But thank you very much, and thank you for keeping to time. We're going to be queuing up Crystal Joinville, who asks actually not to be recorded. So we're going to pause this recording now so that Crystal can give her presentation. But if there's any last questions. No. All right. So then, Crystal, welcome. We're excited to have you. Crystal's presentation is bioaccessibility of cadmium and zinc in wheat seed. And I'm going to stop recording now. And the reminder is just please keep it to 10 minutes, and so that we'll be finishing at 1:10. I'll give you, I might break in at 1:09 or send you a chat at 1:09 to say, okay, wrap it up. And then the other reminder is to everybody who's listening, if you have questions as the presentation is going, just like you would if you were taking notes, put them in chat, right? And then you'll have questions when we finish at 1:10, I mean at 2:10, you'll be able to say, here's my question right away. So if you have questions as Crystal's giving her presentation, why don't you put them in chat, and then we'll be able to speak them at 2:10. And so here we go. Thank you.
Hi, everyone. Can you hear me?
Yes.
Okay. Hi, everyone. My name is Crystal.
All right. Good job, Crystal. And hello, Anaya.
Okay. I'm trying to make this screen. Okay. Okay. Hello, everyone. My name is Anaya, and I am also an undergraduate sophomore, and I work in Dr. Synchronous lab with Crystal as well. So I'm doing bioaccessibility as well, but mine is on rice. And like Crystal was saying, bioaccessibility is basically how much of a substance can be absorbed into the human gastrointestinal tract. And she was running over that cadmium is a very toxic element. It's also found in the soil, but due to human mining processes, the concentration of cadmium has increased over the years. So this becomes a problem when agricultural crops like rice and wheat grow in the soil and they are exposed to the cadmium contaminated soil because people eat them. So if people are eating these crops that are grown in these contaminated soils, they have a potential risk of being sick. So for this experiment, if you're wondering why rice is chosen, rice is one of the top cereals amongst like wheat and corn that are commonly consumed by people every day. And it's been increasing from up to 486.2 million tons per year. So I think it would make sense to do research on something that's been commonly eating and it poses a risk to human health. And another key point that Crystal also brought up was that it's important to look up the bioaccessibility of something like this, because just because the plants are grown in cadmium and are exposed to cadmium, it does not mean that the cadmium that is in the plant will be
Like if it is a high amount of cadmium or a low amount of cadmium, it does not mean that that high amount of cadmium or low amount of cadmium will be directly accessible to the body. Yes, so for this experiment, we use 3 strands of rice, and they'll be referred to as 428. And that in the charts is going to be the color blue. 546, which is going to be orange, and 667, which is going to be green. And in these plants, when they were grown, they were treated with either 0 cadmium or a 1 micromolar solution of cadmium sulfate. They were harvested at different stages of growth. The first stage being anthesis, the second stage being 2 weeks after anthesis, and the third stage being 4 weeks after anthesis. And when the seeds were harvested, they were ground up to mimic chewing, like when you're eating the rice. And then they were further processed with incubation and solutions that mimic the conditions that occur in the stomach and intestine when your rice or food is being digested. So at the end of these processes, the supernatant was collected and was measured with ICP-MS for the bioaccessibility results. So if we go to our results section, we see 2 graphs, and the first graph is of the cadmium concentration found in the rice plants at the different stages of growth. So like I was saying, the blue is the 428 line, and I'm gonna focus on the last stage of growth. So in the last stage of growth, we can see that the 428 line has the highest amount of cadmium concentration, and in the middle is the 667 line, and the lowest concentration of cadmium is the 546 line. And when we go to the graph that shows the cadmium bioaccessibility of the strains, we see that 667 has the highest at about 14%, and 428 has the lowest at about like 2%. So when we look at both of these graphs, we see that the highest cadmium concentration is in the 428 line, but when we look at the bioaccessibility of it, it's actually the one that has the least amount of bioaccessible cadmium. And then also in the concentration graph, we see that the 667 line is in the middle of being the most concentrated, but it has the highest amount of cadmium that is bioaccessible. So from these 2 graphs, we can get the idea that the cadmium concentration that is found in the rice seeds is not directly related to how much cadmium that will be actually bioaccessible to the human GI tract. So yeah, that's like the big picture that we got. And this is also important to note that this information, that cadmium concentration and bioaccessibility not being related to each other, is specifically for these strains. And that would make sense because in Christelle's lab, we see that it's the opposite, where the concentration and the bioaccessibility of cadmium in her wheat plants actually do correlate with one another. So I, along with Christelle, will be doing further research to see how cadmium reacts towards other minerals, like if it increases or decreases and that sort of thing. But yeah, that's the end of my presentation, and I would like to give special thanks to Dr. Senkman and Michael Tavares for helping to support me during this research.
That is fantastic. Thank you, Naya. Thank you very much for that presentation and for that important research. Do we have any questions? I'm going to look over in the chat. Does anybody have questions or clarifications or follow-up they want to ask Naya?
I just want to say thank you also. Really super interesting.
Yeah, it really is. All right, then thanks. Great job, Naya. And I look forward to seeing the, you and your colleagues, your other students work next year, right? As you do the next rounds of these research projects. It's really exciting to think about. Our next presenter is Katie Archer, who's presenting on the familiarity of voice. That familiarity of voice facilitates memory for accented voices in the psychology department. So, and Katie gave consent to be recorded. So we're just gonna keep the recording going, and I'll set the time for 10 minutes from now. So by 2:25, Katie will be wrapping up. And again, if you have questions, put them in chat as you're listening or jot them down. And then at 2:25, we'll ask some questions. Thanks very much. So here's Katie.
Thank you. Hi. Let me just set up the share screen. Oops. Hold on. Okay. So I looked at voice familiarization and how it affects memory in a word recognition task. Familiarization is a process where participants are repeatedly exposed to the same voice. During this process, characteristics of those voices, such as talker identity, pitch, and accent, are encoded. And past research has found that voice characteristics are encoded and used to aid perceptual and memory processes. In previous studies, they found that being familiarized with a voice aided in the perception of novel words in a word identification task, and both of those studies familiarized participants over several days. And they found that the more familiar they were with the voice, the faster they were at identifying the words. But those studies looked at perceptual processes, and I wanted to look at memory processes. So Drozdova and all looked at whether those voice characteristics could aid memory in a word recognition task. And they found that, again, the more familiar they were with a voice, the better they performed on a word recognition task. The familiarity training periods that have been used have ranged from 2 to 9 days. And the varying lengths of familiarization training led me to wonder if it could be done in a shorter time period. The main focus of my research was, can a shortened familiarization period improve word recognition? But I also wanted to look at whether this shortened familiarization period would improve word recognition for accented voices. This was more exploratory though, because there was not a lot of evidence to suggest that familiarity would improve word recognition for accented voices. There was some research on how quickly we can adapt to accented speech. 2 studies found that we can adapt within a minute for a word identification task, but there was no evidence that familiarization benefits accented voices in the same way. So I just wanted to look at it since we had the accented voice available. We were only able to run 8 participants before the shutdown. There are 7 female, 1 male, 3 monolingual, and 5 bilinguals, and the ages ranged from 18 to 55. We ran a power analysis assuming a medium effect with a 0.05 alpha, which revealed that for 80% power, we would have needed an N of 34 in order to find a significant difference between pre and post recognition scores. And the experiment was coded in E prime using 3 voices from Campanou and Elaine's 2019 study. The 3 voices were all male. 2 of them were unaccented Canadian voices that we labeled voice A and voice B. Voice A was our untrained control voice, and voice B was our trained unaccented voice. And the last voice was a Chinese Canadian accented voice, which was labeled voice W. And there were 3 phases to the experiment. The first part of the experiment was the pre-study test phase, and participants listened to all 3 voices in this phase. There were 3 buffer words at the beginning and end to minimize primacy and recency effects, and then they heard 24 study words, 8 in each voice. And in the second part, they heard the 24 study words from before and then 24 new words. And while listening, they had to indicate on the keyboard if they thought the word was new or old in this second part. After this, there was a 5-minute break before they went on to the familiarization phase, where they heard 120 words using only 2 of the 3 voices, voice B and voice W, 1 accented and 1 unaccented voice. There were 60 words in each voice, and while hearing the voices, they had to type the word that they heard. After the familiarization phase, they did another study test phase identical to the first one. And in each phase, we used new words, and the lists were randomized for every participant. My main prediction was that performance for the familiarized unaccented voice would show greater improvement from pre to post than the untrained voice. I thought there could be some improvement pre to post for the untrained voice, but that it wouldn't be significant. And I also predicted that there would be greater improvement for the accented voice, since we would start at a lower performance level because it was more difficult to process. And since we adapt rapidly to them, we would show greater improvement overall. We ran a 2-factor repeated measures ANOVA of the hits minus false alarms for each voice pre and post familiarization. And we found there was no significant effect of time or voice, but there was a significant interaction. When we looked at voice alone, we found that the only significant difference among the voices was between voice B and voice W, which were our trained voices. Overall performance for voice B was not significantly different than voice A, and the difference between voice A and voice W was also not significant. Voice W overall was significantly better in performance than voice B. When we looked at time, there was an increase in performance from pre to post overall, but it wasn't significant. This analysis included all 3 voices, and we know performance improved for voice A, which was not used in the familiarization phase, and that indicates that familiarization was not a factor in their improvement. We also looked at the interaction, and we saw that there was a significant interaction, which was driven by voice W. Performance for voice B improved from pre to post, but it got worse for voice W, and this change in performance was significantly different, and that's what drove this interaction. There wasn't a significant change in performance between voice A and voice B, or between voice W and voice A, but it approached significance. So did the familiarization work? Well, looking at the trained non-accented voice, there was no significant main effect, but there was a trend in the expected direction. Performance for voice A, which was untrained, was overall higher than performance for voice B, but the slope for voice B was steeper. This could suggest that the voice B was improving, performance for voice B was improving more than the untrained voice, but since it wasn't significant, that's only speculation. Since there was a difference pre to post anyway, I can't say that the familiarization had an effect because the main effect of time was not significant. My second question was whether this shortened familiarization phase could work for accented voices. And again, this was an exploratory question since we had the accented voice just to see if it would have the same effect. Based on this, we know that there was a main effect of voice and a significant interaction. And the interaction shows that a shortened familiarization period does not work for accented voices because the trend was in the opposite direction. They did worse after the familiarization. We had a very small sample size and low power. So if we'd had time to run more participants, we may have seen an effect of the familiarization for the unaccented voice, since there was a trend in the direction we expected, and a steeper slope for voice B than for voice A. Another limitation was the person who recorded voice A had better pronunciation, but we can look at the difference in slopes to see if there was an effect of the familiarization. But in future studies, we would try to minimize this difference if possible. As for why we got these results, it's possible that the length of familiarization, oops, that's too far, sorry. It's possible that the length of familiarization that we chose is not long enough to have a significant effect on performance in word recognition. Previous research had found that a significant effect could be achieved in as little as 2 days, but it could be that a longer familiarization is needed for a word recognition task. It's also possible that this wasn't long enough for the listening condition we chose. In the previous studies, they found that people rely more on voice characteristics when the stimuli is presented in noise, and we presented them in clear listening conditions. As for performance for voice W, it was initially higher than for the other two voices, and we saw an unexpected decline from pre to post. And one theory that I have is that there was an initial boost in performance for the accented voice because it was unfamiliar and more difficult to process. So participants may have devoted more attentional resources to that voice since it was more difficult to process. Then as they became more familiar with the voice, they didn't need that attentional boost as much, which could explain the dip in performance post-familiarization. It's possible that familiarization works differently for accented voices, which is why we're not seeing the same trend as we saw for unaccented voices. And this attentional bias could be part of a different mechanism at play for accented voices. There's evidence that we adapt rapidly to them, but that rapid adaptation may not add to an improvement over time once we've adapted to the voice, but rather just an initial boost. I would suggest that future research look at rapid familiarization for unaccented voices using a larger sample. Since our sample size was so small, we don't know based on this study that it couldn't work. And I would also suggest using different listening conditions to see if adding noise or different levels of noise can impact the performance level for participants. So hopefully that's something we can look at in future studies. Thank you.
That's wonderful. Thank you very, very much, Katie. Does anybody have any questions on this presentation? I was really fascinated by it. I think it's one of the great strengths of Lehman and living in New York is that we, we listen to people with all kinds of different voices and all kinds of different accents and the many different languages feeding into the accents that they speak English with. So I'm sort of fascinated by the research, and especially on what I think you were saying towards the end of your presentation, that maybe we devote more attention when we're having to listen a little more carefully, and therefore we might remember a little more on an accented voice. I think that's really cool. I think that's one of those perfect New York extra strengths, Yes. So thank you for that research.
Thank you.
We have Anton Kirilenko coming up next, who's going to present on the semi-analytic framework for studying the performance of the second generation Extreme Universe Space Observatory on board a superpressure balloon.
Hello, everyone. Can anyone hear me? Okay, good. All right. Okay, so I'm Anton, a current junior at Lehman. I'm doing research in the Department of Physics and Astronomy with Professor Encardoki and his graduate student, Jorge Soriano. And the project that we're currently doing on is based on a semi-analytic framework for studying the performance of a cosmic ray telescope that's going to be suspended on a superpressure balloon and sent up high into the atmosphere. So introductions, in order to study the performance of a space-based fluorescence cosmic ray telescope and be able to send it up into high in the atmosphere, we first have to do a full simulation of the showers, fluorescence production and propagation, and detection at the telescope must be performed. So, however, a lot of this stuff and to do a full simulation, it requires a lot of computing power, and it's very hard to access a supercomputer for most of the universities. However, we can also obtain a lot of useful information for the project by using a simplified framework based on analytical calculations and numerical integration of parameters set about the atmosphere and the gravitational field of the Earth. So, and a lot of that knowledge has already been made before, so you can use the previous accumulated information about the Earth for this study. So in this talk, I'll present a new scheme based on well-established techniques that attempt to study the average response. So average meaning that you take well, like an average response and take like statistical data on how the showers develop in the atmosphere in order to send the space balloon later to detect cosmic ray showers. So before doing that, the framework there, I want to first say what are ultra high energy cosmic rays. They're basically highly energetic subatomic particles, mostly protons, which is probably around like 99% of all the cosmic rays. However, it can be heavier nuclei such as metal ions and other ones as helium and alpha particles. And basically they travel at essentially the speed of light. And from theory of special relativity, you can't actually have a particle reach the speed of light. However, these particles travel at such a high speed with so much energy that you would have to put 99.9 and then 100 more nines after the decimal point to describe how fast these particles are going. And the energy for these protons are basically 10 to the 18th power electron volt, which are units used in particle physics to convert easily between mass, energy, speed, and time. And so ultra high energy cosmic rays collide with the atmospheric nuclei, mostly nitrogen atoms, since around 70% of the atmosphere consists of them. Of those atoms, and when they collide with those nitrogen atoms, the interactions produce electrons, mesons, and gamma rays, which are all, except gamma rays, are all particles in the standard model of physics. So these secondary particles then react with the more air atoms, producing a propagating cascade of particles that can be detected by the emitted UV fluorescence light. And that's what we call cosmic ray showers, essentially. And if you look at the graph to the left, this shows the average flux of these cosmic rays. And if you look to the top left corner of the graph, the lower energy cosmic rays have a larger flux, so they come into the atmosphere much more often than the ultra high energy ones. And just how rare those events are is basically estimated to be 1 event per square kilometer per century per steradian, which is basically in simple terms is very rare. It's very rare occurrence to have these cosmic rays enter. However, they're still detectable. And the image I have of Serena Williams is basically to show that the highest energy cosmic ray particles have an energy of tennis ball served on average by Serena Williams. So although it might not seem like it's a lot of energy, if you compress it into the size of a proton, it will launch it basically at the speed of light. And I did the recent calculation based on it to draw another scale of how much energy these particles have. So if you were to take 1/10 millionth of a gram of these particles and put them together, it would equal to the energy of a nuclear bomb. So that just kind of says how energetic these particles are. So the origins of these cosmic rays are mostly of extragalactic origin. Possible sources include galactic nuclei, dormant quasars, and the supermassive black holes. And you can you can look up more info, which was done by Claire Meckman in the talk earlier, I think. So these particles often go past the theoretical limit, which is known as the GZK limit, which is the 5 to the times 10 to the 19th electrovolts. So at energy is greater than this limit, we would assume that the cosmic rays start interacting with the radiation of the cosmic wave, microwave background, and they would lose energy. However, this doesn't necessarily happen. And to the image on the left, you can basically see that these particles come out a stream of jets from, let's say, this example, a supermassive black hole, and it's assumed that it's one of the few sources of generating so much power for a single particle.
Just a quick reminder that you're just about close to time.
Okay. So detection of ultra high energy cosmic rays, it's important to know the arrival direction, arrival time, intensity, wavelength of the fluorescence light. And once these properties are well understood, we can basically send up a space telescope on a balloon. The physical processes involved in the arrival of the cosmic ray to the Earth's atmosphere involve the primary interaction of the cosmic ray, evolution of the shower, the fluorescence production, and the propagation of the fluorescence through the atmosphere. The physical model can be divided into the atmospheric model, shower development model, fluorescence model, and the optical one. So the atmospheric model that I did basically set up the Earth as a geometric system, and based on it, using the geopotential altitude and other parameters, I was able to set the ATM temperature, pressure, density, ATM number density for the Earth in order to have the density known of the nitrogen atoms based on the altitude. And continued on with the atmospheric model, it just goes on to calculate the humidity through other various equations, which are important to know. The shower development model is again determined by those previous atmospheric properties, and it produces a function quantifying the energy deposit per unit length along the shower track. And since it's a semi-analytical study, we're interested in the average response of the detector. All right. And the fluorescence module basically expands on the previous propagation model. And as I was saying before, the fluorescence comes mostly from the de-excitation of the nitrogen atoms in the atmosphere, and humidity plays a role in how it affects the peak and wavelengths, as was described in the graph here. And the science goals and future work is to identify the origin of these cosmic rays, measure the energy spectra, and improve the knowledge of particle physics. So improve on the standard model. And the future mission is basically to send a full space telescope without a balloon and into orbit. Thank you.
Thank you. Thank you, Anton.
Sorry if it was, I don't know if it went overtime.
Only a little
I
bit. No, but
miscalculated about it.
No, you're okay, just a little bit.
Yeah.
But
it always happens to me, to be honest. Once I start presenting, I just go over time every single time.
yeah, no, it's great. You're into the research. You've done incredible research and you know a lot about it. So good job. That's lovely. Who has any questions?
I see lots of Zoom claps. That's good news.
Mm-hmm. Yeah, exactly.
Well, perhaps a typical question would be like, what's the purpose of it? Is basically is to improve on the standard model of particle physics. So some of the stuff that we do, let's say in a particle accelerator, is to detect new particles such as the Higgs boson and new pions and all that stuff. And cosmic rays are one of the possible sources of delta plus plus baryons, which are just one of those particles that we could add to the standard model and just goes as like a whole foundation for the whole particle physics and chemistry and beyond all that stuff there.
Right.
Yeah.
Yeah, exactly. All right. Well, thank you. We're going to hear, I'm sure you, like all of these other presenters, are going to continue doing really interesting research, right? Like really fantastic and kind of barrier busting scientific and sociological and psychological and all kinds of research. It's really fantastic. So thank you very, very much, everybody who's presented so far. And we just have a few more people to present today. Weldy Guzman and Sheila Devia are going to present on persistently luminescent nanoparticles. So you are up, Weldy and Sheila. And you'll speak from now until 2:50. And again, think up questions, people, as you're listening, and then we'll be able to go. And we're going to pause the recording because they asked not to be recorded.
Zoom the recording now, and then Eileen, will you also resume on your end and then stop when it's done?
Mm-hmm.
And we'll be able to upload Anton's slides with the rest of his presentation. So thank you all very much.